BioMindscape. Here the question is, during the chemoosmotic ATP synthesis in the chloroplast, there is a high concentration of proton, that is H+, and it builds up inside the thylakoid lumen. Which one of the following process does not directly contribute to the proton gradient? We have to tell which one does not contribute to the proton gradient. So here A is splitting of the water molecule on the inner side of the thylakoid membrane, B is the reduction of NADP to NADPH on the stroma side of the membrane, C is conformational change in the F1 particle of ATP synthesis, and D is the movement of electron through the cytochrome B6F complex pumping proton from the stromata. So here, which one is the correct option? So here the correct option is the conformational changes in the F1 particle of the ATP synthesis. Means this one is an incorrect option. Option A is the correct we will mark because it does not contribute. Water splitting complex is located on the inner side of the thylakoid membrane. When the water split, the proton are directly released in the lumen, increasing the gradient. Whereas option B and D, they also contribute in this NADPH reduction. It happens in the stomatal side and it removes the proton from the stomata, making the stoma less acidic, while it increases the gradient between the lumen and the stoma. Whereas option D, we can see the cytochrome complex, it acts as a protein pump. It actively moves the proton from the stomata into the lumen. So your correct option is C. Next question is a biologist introduced an antibiotic into a bacterium culture, and the antibiotic binds specifically to the 50S ribosome subunit and prevent the movement of ribosome along the mRNA strand. So which specific step of translation is directly blocked? Option A, amino acid activation. Option B, initiation, complex formation. Option C, peptide bond formation. And option D, translocation. Here the option D, that is translocation, it is a specific process where the ribosome move down the mRNA strand by one codon distant. Thanks for watching. Do like, share and subscribe with your friends.